All right, so here I have on the screen an operon. This is just one of many models. In this operon model, let's just review what we have here. I have my genes that are going to be transcribed and translated into a protein. That protein at the end of this is going to be something called tryptophan. So tryptophan is something that's very important for us to build things. So this is a building, this operon is going to produce a product that's going to allow us to build something, tryptophan. I have my operator, which is acting as my on-off switch. I have the promoter, which binds the RNA polymerase. The RNA polymerase is the enzyme responsible for the polymerization or the transcription of the DNA. So those are the main parts of this. And then somewhere upstream, which is not really relevant to my conversation, is the regulatory gene that's going to code for the repressor. Here, I have the repressor. Okay? That's my repressor. So, by default, let's just get rid of these things here. I should have started with these off the screen, sadly. All right. So by default, my RNA polymerase is floating around the nucleoplasm and it gets attracted to the promoter. We know there's a bunch of transcription factors that are going to bond and allow the whole process to go much more quickly. I'm not going to talk about that. You already learned that. So the RNA polymerase bonds to the promoter. And then you'll notice that it's free, nothing's stopping it from moving. It's free to move down the DNA past the operator to gene 1. At gene 1, this is now going to transcribe. I'll use green here. It's going to transcribe, start transcribing. And it's going to keep on moving along. Gene 1, gene 2, gene 3, gene 4, gene 5. And there's a couple ways this can happen. But at the end of this, what's going to happen is you're going to end up with, right? Remember, I was kind of skipping steps here. Right? So the end of this, you're going to get the transcription and translation of 5 proteins. And the RNA polymerase has moved off, it's done its job, done. These proteins ultimately come together and they're going to produce tryptophan. So at the end of this, tryptophan is produced. And the tryptophan is going to continue to be produced as long as the RNA polymerase is able to bond and move. Bond and move, right? There'll be a number of different RNA polymerase, bond and move, right? So I end up with all this tryptophan. So the question becomes, when do you stop producing tryptophan? Because at some point you have enough tryptophan. You don't need to produce unlimited supplies of tryptophan. You'd be burning through energy all the time. Your body's incredibly efficient. So it's going to produce enough tryptophan. And you need some kind of mechanism to shut this down. And so we have that mechanism. That mechanism is the tryptophan itself. When there's enough tryptophan, the tryptophan will act as a co-repressor. And it will bind to the repressor molecule. And when it binds to the repressor molecule, it will transform, right? The repressor molecule is a protein. This is binding to the allosteric site, causing a transformation of the active site. So this whole molecule is going to change shape. And as it changes shape, it's going to change shape into this form that now fits the operator. And now that this repressor with the co-repressing tryptophan combination binds to the operator, it effectively turns it off. And the next time the RNA polymerase bonds, it is no longer able to move. It's impeded by this repressor. It is now turned off. And that is why the trip operon is known as a repressible operon. Okay, we call this a repressible operon. Repressible operon. And one of the key features of a repressible operon is they typically function in an operon or a pathway that is, well, you tell me, is it catabolic or is it anabolic? Is it building or is it breaking down and producing energy? In this case, we're building, right? We're building tryptophan. So this is what you typically see in an anabolic pathway, repressible operons. And the end product is what's going to cause the operon to be turned off, okay? Now, as a tryptophan gets used for whatever purpose, building some kind of protein, some other bigger protein, other processes, and those all get used up, then, let's take them off the screen here, then what will happen is this tryptophan will disassociate. and it will no longer bind to the repressor, and it will be used for something. And when that happens, it causes the repressor to change shape. And so as it changes shape, it now comes off the operator, and the RNA polymerase is free to move. And at the end of this, we end up producing tryptophan again, and more tryptophan, and more tryptophan, and that is the trip operon, a repressible operon. So that's operon number 1. You can go through the PowerPoint notes and make sure you take specific sequence notes of how the process works, and then come back here to see the teaching of the process. That brings us to the lac operon. Just by looking at it, you can see this is a more complicated operon. That last one is straightforward. We had the RNA polymerase, it went down the line, it produced something, and when there was too much of it, repressed it, turned it off. Now take a look at this operon. The lac operon is responsible for allowing the prokaryote to metabolize lactose. As you know, lactose ending in OSE is a carbohydrate. Carbohydrates are metabolized to produce energy. So lactose is a source of energy. Not the best source. The best source is glucose. But if glucose is not available, then other sugars can take its place. So the lac operon is going to allow the cell to metabolize lactose and produce energy in the form of ATP. So let's review the structure of the lac operon. The lac operon has 3 major genes, structural genes. It has LAC-Z that produces beta-galactosidase. It has LAC-Y gene that produces permease, and LAC-A that produces transacetylase. The beta-galactosidase is a protein, an enzyme, that will break down lactose into glucose and galactose. That's its job. So that's a great first step. You got lactose, you got to break it down into monosaccharide, into glucose, and you get energy. Permease allows lactose to enter the cell, so it makes the cell membrane permeable, allowing lactose to come in. Transacetylase is not important for our purposes, but it's structured, it's packaged with this operon. So if permease doesn't work, lactose can't get into the cell, and therefore you can't get energy from lactose. If beta-galactosidase doesn't work, then therefore the lactose is not broken down into glucose and galactose, and you don't get energy from lactose. So both of these are very important. Take a look at my operator. You'll notice to my operator I already have the repressor bound. In the lac operon, the repressor is bound by default, unlike the trip operon where the repressor is not bound by default. And then the promoter, I have something weird going on here, right? The promoter, I got a special little piece that's kind of missing here. We'll talk about that in just a moment. So if this RNA polymerase binds the lac operon at the promoter as it's supposed to, by default, will this RNA polymerase transcribe and translate? Taking a look at my model, you should say an emphatic no. Hopefully you guys said that. All right, so the RNA polymerase does not move. That's fine. You want this model, this operon, to be turned on under 2 conditions. And one of those conditions is you need to have lactose present. If there's no lactose present, don't turn on this operon. It makes no sense. It's a waste of energy. So one of the very first things that we talk about is turning this on. It so happens that lactose, I'm going to use different terminology here. Often people will call this an inducer because it induces transcription. But technically, it's actually a repressor, a repressor of the repressor. It bonds to the repressor, the allosteric site. It causes the repressor molecule to change shape so it's no longer bound. Right? So once this lactose binds to the repressor is going to change shape. So let me move it off. I'll dump it behind this so you can't see it. I'll send that back. Order, send it back. And once it changes shape, it's going to leave the operator. And now the RNA polymerase is free to move. And it produces LAC-Z, LAC-Y, and LAC-A. And the lactose now can be brought into the cell and broken down. Everything's good. Right? Produces my LAC-Z, my LAC-Y, and my LAC-A. Produces my beta-galactosidase, my permease, and my transacetylase. So we're all good. But here's the other problem. I said there's 2 things the lac operon should have, really. One of them I mentioned was lactose. That totally makes sense. Now, which energy source is better? I mentioned this already as well. glucose or lactose? Clearly glucose. It requires much less effort to take glucose and get energy from it. We did a whole unit on that. If there is glucose available, let's use it. So if there's glucose or lots of energy, let's use that energy. The only time we want this being used is A, if there's lactose, check, and if the energy levels are low. What molecule is in a high abundance if there's lots of energy? That molecule is known as ATP. We've talked about that already. So there's lots of energy, there's lots of ATP in the cell. ATP is correlated against AMP, right? There's ADP for adenosine diphosphate and AMP for adenosine monophosphate. So if there's lots of ATP, then you'd expect the CAMP levels to be low. And if there's not a lot of ATP, you expect the AMP levels to be high. So the cyclic AMP is a messenger, a signal molecule that tells the cell what the energy levels look like. Okay? So if there's not a lot of ATP, there's a lot of CAMP. If there's a lot of ATP, there's very little CAMP. So let's say that there's lactose. Great. Lactose binds to the repressor, pulls it off, the RNA polymerase is ready to do its thing. But the promoter here is not complete. So the promoter will still attract RNA polymerase, yes, but not very strongly. So you need one more thing to happen. And that comes from CAMP. So if the ATP levels are low, that means the CAMP levels are high. If the CAMP levels are high, then there will be enough CAMP to bind to this other very special protein complex. And together they form something called the CAMP-CARP complex. Different books have different names for this complex. It's a complex. And together, this will bond to the promoter. And when it bonds to the promoter, it forms a complete promoter. And this promoter is now able to bond the RNA polymerase effectively. So that is the second level of control. The first level of control is the lactose being available. The second level of control is the energy level being low. Now that both of those have been met, the RNA polymerase rockets to the promoter. It then moves down past the operator, which is free, and it will transcribe and translate the LAC-Z, LAC-Y, and LAC-A to produce those enzymes. Done. So those are the two mechanisms that allow the LAC operon to work. One of them is a negative feedback, and one of them is a positive feedback. I'm specifically going to just say those words, and I'm going to ask you to review the terminology. And then in the live class, you can ask me or clarify for me what is negative versus positive from the PowerPoint. So I'm asking you, without me talking about it specifically, can you tell me which one's positive and which one's negative regulation? That's a big theme in our bio course, positive and negative regulation. This is control, right? You'll see it in genetics. You'll see it in homeostasis. You'll see it in metabolism. You see it all over the place. All right. So if the energy levels begin to get very high, right? So the ATP starts to build up as you get more energy, then this CAMP will start to get low. And then what's ever attached to this CARP, it will disassociate. As it disassociates, the CAMP-CARP complex will leave the promoter. And then the RNA polymerase will not be as effectively bound to the promoter, and the whole process slows down. All right, so that brings me to, I guess, the last stage. And this is where we combine the genetic control with mutations. In the actual activity set, that document I gave you, at the beginning of the document, I outlined what the concepts are that you should know. Mutations, with frame shift and point being primary, and that Word document with the worksheet, it's right here, is a good exercise. The DNA repair, which you're learning from the website by clicking on the link. The control, which I just talked about, specifically the LAC operon and the TRP operon. In the PowerPoint, you'll see that reference to the LUX operon. Go ahead and click on it to feel that, there's lots of operons. You have a bunch of resources to help you, but as you jump down and you finish this operon, if you think you understand it, and I encourage you to work with somebody, don't just work by yourself. You're going to look at the fish operon. The fish operon. So let's just go over this together. This is your activity, part of your activities for the week. You're going to submit this, and it'll be check or incomplete. So in the fish operon, there's this crazy looking when I was younger, the shows were not as sophisticated. And one of the shows that we watched was this guy named Elf. Elf was from the planet Melmac, right? So Elf, alien life form, that's what ALF stands for. And he's from the planet Melmac. So this was the kind of alien show that we used to watch, Elf. And go to images here. And there he is, Elf. And Elf had this thing for cats. So if you know the show, and the next activity would make a little bit more sense to you. Anyway, there's Elf, cool guy. He's totally misplaced. He thought he was smarter than the rest of us humans. I guess he was. I mean, he got here. We're still here. And on the planet Melmac, there's this operon that allows Melmacian cats to metabolize fish. So let's take a look at it. You have the structural genes of F, I, S, and H. And each of these structural genes does something very specific. It tells you gene, 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 and G. So there are 4 specific structural genes. T is a terminator. So I didn't mention that, but the terminator is where the instructions for the RNA polymerase to leave the DNA are set. The operator is the on-off switch, as we know. And then the promoter, here is the promoter. Here's that cap sequence, which I kind of described with the lac operon. And then here's the I gene that codes for the repressor. And here's the promoter for the repressor, because the repressor needs its own set of, control elements. So the repressor has its own promoter. And this here, cap and go fish, are the promoter for the fish structural genes. So you're going to work your way through this. To understand the questions here, you do need to know the difference between a stimulus, something that makes something happen, and a symptom, an indication that you have the malady or whatever it might be. This little diagram here, that's supposed to be a point. So it's just a bad edit on my part. That should be a triangle on the top of this little molecule. Okay, so that's your fish operon. Work through that, work with a partner, submit it, and make sure that you understand the process through that document. It's supposed to be fun. It's kind of complete or incomplete. Okay. Mutations. So what are the mutations that can occur from the lac operon? Let's start with the super repressor. So here's a repressor that's bound. And I'm going to mutate this repressor. So I'm going to go up to the regulatory gene. I'm going to do something that's going to mutate it. And it's going to cause the repressor to look like this. This is known as a super repressor. And we designate it IS, or super repressor. So what's happening with my super repressor? Well, can my lactose bind to the super repressor? And the answer would be no. It can't bind because there's no allosteric site to put this, There's no allosteric site to put this on. This can't bond. So because it can't bond, that means this repressor will never leave the operator, and this will be turned off permanently. The RNA polymerase will bond, and it can never make it past, right? So the super repressor, it will permanently turn off the operon. So that's one type of problem. Okay. Next, we can have an operator, right? So I know it's the wrong color, but the operator, so we call this one OC, right? OC. So this is an operator mutation. And in this mutation, the operator does not have a site that will bond the repressor. So the repressor, it can't bond. There's no place for it to lock on the operator. So since the operator will not bond this repressor, that means this operon will never be turned off, right? So it's on all the time because the repressor can't bind. So that's another type of mutation that can occur. All right. Another one that can occur could be an I where the repressor is broken, right? So the repressor, we call it a negative. So that's typically a defect that turns off that protein in some way. So if that were to happen, I might have a repressor that looks like this, right? So now this repressor here, it won't bond because it doesn't have the right site to bond to the operator. So that I negative repressor, another mutation, this would be permanently on. And then my final mutations are structural mutations. In my structural mutations, that's where your Z gene might be affected. So if the Z was not working properly, what would that mean? Well, it means that the beta-galactosidase would not be produced properly, and therefore the lactose wouldn't be broken down, and therefore you wouldn't get energy. Or the permease might be affected. If that happens, that means the lactose can't even make it into the cell, and therefore you can't use lactose to break it down, and you can't get energy. So we have several different types of mutation. We have the super repressor I mentioned, so that's permanently off. We have the operator mutation, so it's permanently on. We have another inhibitor where it's defective, so it can't bond, so it's permanently on. And then we have the Z and the Y. So we have several different types of mutations. So these are operon mutations. So that ties the mutation story to the genetic control story, bringing it together. You'll see a multiple choice worksheet with answers, solutions, I guess we'll call them, that you can work through on the site for practice. That brings to an end our, wait a second, where's my document? That brings to an end our practice. So as you look at this document, you'll see there's going to be a number of things. I didn't update it from the website. Mr. Desange put some things up there as well. There's a Learn 360 resource that I would suggest you do right after the mutation practice. There is the fish operon down below. There is the end of the week quiz, which we're going to have on Friday. And then we have a competition on Thursday. And starting from now on, we're going to have these question competitions. There's going to be a series of questions. It's going to be kind of like a quiz, except it's going to be competitive. So you guys are going to compete against each other to see how fast you can answer the questions, as well as how accurately you can answer the questions. So it's like we've done it. My class, we've done these quizzes before, so you're familiar with it. And anyone in HOSA, you've done these as well. Okay. There'll be a couple of other resources up there as well. One is a PhET simulation, and the other one is a movie. And that will help prepare you for the biotech. And we'll talk more about the biotech later this week. So we're going to give you some information about your culminating task for this unit later this week so that you guys can start watching movies and preparing some stuff for it. I can't tell you too much because I'm at 22 minutes and that's way too long. All right. Thank you, and I'll talk to you soon. Remember today, Monday, I'm not sure when you guys are listening to this. You might be listening to this after I've already talked to you. Sorry, not today. On Wednesday, we're going to have a live class, and that's supposed to happen at 2 o'clock, I believe. And I'll send a link where we'll all meet. We'll have a conversation. We'll talk about the culminating task, which will be due the following week, and we'll talk about any questions. The reason I'm having it Wednesday, because there's often questions around the operon and truly understanding how it works and getting an explanation as to why you got questions wrong or whatnot. So Wednesday we'll give you an opportunity to get started on this. The mutation stuff's super easy. It's just memory work. It's the operon where you really need to spend the time on so you can ask good questions. Okay, thanks and goodbye.